Give you guys a minute to knock that out. Say that one more time. For tonight's homework? All right, let's see. So its due date is the 9th. You said when is the homework due? Yeah? It says due date is the 9th.
Some plants have like fur on them.
It usually has to do with the defense mechanism. It's usually poisonous.
Oh, okay, that's kind of scary.
All right, guys, a couple more minutes. We're going to get started. All right, so let's first start by going over what a homologous chromosome means. Again, what does homo mean? Same. So remember when I said that you get half of your chromosomes from your mom and the other half from your dad, right? And so homologous chromosomes are the same size. Go ahead and write this down. They're the same size, and so we match them up. So you get one of those chromosomes from your mom and one of them from your dad, and they carry the same genes on them. The difference is that those genes could be varied. So your dad might have given you the gene for blue eyes, and your mom could have given you the gene for brown eyes, but it's still the gene for eye color. Does that kind of make sense? So body cells are called somatic cells. Please highlight somatic cells. You're going to need to know that. Those are your regular body cells, like your skin cells, the cells in your eyeballs, your brain cells. All your regular body cells are called somatic cells. So highlight that. They have 2 of each kind of chromosome. Again, one from your mom, the other from your dad. So when there's 2 of each kind of chromosome, we call that diploid. Di means 2. Di means 2. So we say it's diploid or 2N. The N being chromosomes. So the opposite of a somatic cell, body cell, would be a gamete. These are sex cells. So what are the 2 sex cells? What are the 2 sex cells? Egg and sperm. Okay, here's the issue with egg and sperm cells. Your egg cells and your sperm cells cannot have 46 chromosomes because your regular body cells have 46, but your egg and sperm cannot. Think about it this way. The egg and the sperm are meant to fuse with one another. And so if they each had 46 chromosomes, that would make a baby with how many chromosomes? 92. And that would no longer be a human baby, right? So your sex cells have to have 23. 23 in the egg, 23 in the sperm. So that 23 plus 23 makes a baby that has what? 46. So somatic cells are diploid. Make sure you highlight that. And gametes are haploid, meaning half the number of chromosomes. Please highlight that. Be reminded that this is also on your Canvas. So if I go too fast, it's there for you to go back and look at. N represents the number of chromosomes in each cell. Okay? So heads up, a human, again, has 46 chromosomes in a body cell, in a somatic cell, right? And 23 in a gamete. A fruit is 8 in a body cell, 4 in a gamete. So it is different in different organisms. A dog has 78 in a body cell and 39 in a gamete. So it is different in different organisms. But all we care about are humans, right? So I want you to draw this where it says diagram. You only have to draw the chromosomes. You don't have to draw the plants next to them. Just the chromosomes. So homologous chromosomes, the 2 chromosomes in a pair in a diploid cell. So you would have gotten 1 from mom, and the other from dad. Okay, so this is, these are chromosomes from a pea plant. And yes, plants do have sex. It's just not as interesting as human sex. But this individual had this chromosome from his mom, or its mom, and this chromosome from its dad. Notice here's a gene, here's a gene. It's for flower placement. So mom's gene has a terminal flower placement, and dad's gene has an axial on the side. But dad's gene is a capital letter. So what do you think that means? It's dominant. Therefore, this child or this offspring is going to have axial flowers. Does that kind of make sense? So it all depends on what's dominant. Again, here is another gene for plant height. Same gene for plant height. The variation that mom gives is the tall variation, and dad gives the short variation. But which one's dominant? The tall, right? And then here's pod shape. Same gene. Okay, so same trait. Mom gives the inflated pod. Dad gives the constricted pod, but the inflated is going to be dominant. So this plant is going to have axial flowers. It's going to be tall with an inflated pod. And that's how homologous chromosomes work. 1 from mom, 1 from dad. They each have the same genes, just variation of those genes. So what we need to talk about is how that variation occurs. How is it that your sister has curly hair and you have straight hair? Like, how did that happen? Shouldn't you have both straight hair or both have curly hair? Why is there a variation? Why is your brother ugly and you're gorgeous? How did that happen? He got the ugly genes and you got the pretty genes. So you don't have to draw this, but I do want you looking. So the first 22 pairs of chromosomes are called the autosomes. And the last 2 are called the sex chromosomes. But write that down. So autosomes they're body chromosomes. Write it down where it says homologous chromosomes. They're the body chromosomes, and they're the first 22 pairs. So you have 44, right? 22 pairs is 44, right? So you have 44 autosomes. They're the regular body chromosomes. So they code for regular body stuff. Eye color, height, Do you have a widow's peak? Do you have a hitchhiker's thumb? Blah, 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 blah. All the regular body stuff. Okay, the last two are called sex chromosomes. Write that down. What do you think they code for? Your gender. Write that down. Codes for gender. Write that down. Codes for gender. Does anybody know the sex chromosomes for female? We are XX. So females are XX. So this individual is a female. And then males are XY. So XX for girls, and then XY for boys. Write that down. XX for girls, XY for boys. That having been said, the only individual that can determine the sex of a child is the male, because only males have an Y chromosome. So each sperm cell either has an X or it has a Y. Every egg cell only has an X. Does that kind of make sense? Because females only have X's. So all those times in the years before when, girls got in trouble for not having boys, it never had anything to do with girls. It always had to do with the male. Only the male can determine sex. Okay? All right, so moving on to meiosis. Okay, some things that you need to know. Meiosis is a two-part cell division process. Results in gametes. Those gametes will have 1/2 the number of chromosomes of the parent cell. So in the end, you'll have half the number of chromosomes. This is different from mitosis, because in mitosis, you end up with the same number of chromosomes. It's like an identical, it's like a clone. Okay, so mitosis, like with your skin cells, they just clone themselves. It's asexual, it's cloning. Whereas meiosis, not the same situation. First of all, we're going to divide twice. So not once, but 2 times. And we're going to reduce the number of chromosomes when we divide. So in the end, instead of getting 2 daughter cells, you're going to get 4. All the daughter cells are going to be haploid, which means they're going to have half the number of chromosomes. So instead of having 46, they'll have what? 23. Because we don't want an egg cell with 46 and sperm cell with 46 making a baby that has 92. That's not human. So it has to be 23 plus 23 makes me. I'm sure you've heard that commercial, 23andMe. Yeah. Now, here's what's really cool. So that variation that makes you good looking and your sibling ugly, that's because there's a shuffling of chromosomes. And we're going to talk about how that works. So there's a shuffling of chromosomes so that each new cell has a unique combination. That shuffling is called crossing over. So today we're going to do a lot of drawing. Okay. Guys, try not to get too far ahead of me because you know I don't make you write everything. So if you get too far ahead of me, you might write stuff that you're not supposed to. Okay, here's what I need for you to understand. We're going to start off with what's called a germ cell. So before an egg cell is an egg cell, it's a germ cell. Before a sperm cell is a sperm cell, it's a germ cell. Okay, so we're going to start off with that germ cell. That germ cell is kind of a normal cell. It's got 46 stranded chromosomes. Okay, so it's got 46 chromosomes, so we're good. You don't have this paper, so you're going to draw. So starting at interphase, you're going to draw it on top and describe it on bottom. Or you can describe it on top and draw it on bottom. I don't care. So we know during interphase, we go through G1, S, and G2, right? And so we grow, and then we replicate chromosomes, and then we grow some more. When we replicate, we end up with 92 chromatids, right? Because 46 times 2 is what? 92. Okay? Kind of draw that off to the side. Chromosome duplication. You get to chromatids attached. And what's the middle called? The centromere. Now we showed that last class, right? Like we did that last time. All right. Now we go on to prophase 1. So here's prophase 1. In prophase 1, the chromosomes become visible, but something else happened. God bless you. And so I'm going to let you guys write that down, and we're going to talk about something else that happens during prophase 1. So we did this last time. Remember when we made our chromosomes double-stranded and they were more condensed, we could see them. So something else happens here, and I want you to write it down in your description. Crossing over occurs. And so I'm going to describe or explain what that is. Write that down. Super important. Crossing over occurs. Crossing over occurs. Crossing over occurs. So here's what happens. Here's the chromosome that I got from my mom, and here's the chromosome I got from my dad. So here we go. Here's that chromosome, here's the mommy chromosome, and then here's the daddy chromosome. You with me? They're double-stranded. They should be connected. Here's the kinetochore. Okay? Now, what happens is that this chromosome crosses over with that chromosome. And it takes pieces off of each other. Okay, so it looks like, and Miss Johnson's not great at drawing, but it looks like this. Where they just kind of take pieces off of one another when they cross over. When they take pieces off of one another, it's totally random. And then they're no longer the same chromosomes they used to be. This is very important. Guys, you need to write this down. I can't tell you how many questions on your test is over this. So that crossing over increases variation. That crossing over of chromosomes increases variation. So they cross over, take pieces of each other, totally random. You don't know which genes are going to get swapped. So it's like a quick little swap of genes. Like, here, you want this? Oh, no, I want that. Okay. We don't know. We don't know which ones are going to get swapped. And in the end, when they're done, they don't look the way they used to. So they don't have the same genes they used to have. So this one, the mommy chromosome, might look something like this, right? And then the daddy chromosome might look something like that, because they've taken pieces of each other off. Like I said, it is totally random. We call that crossing over, and that happens during prophase 1. Make sure you write that down. Any questions about that? We good? All right, I want to make sure we're good on that. So that is one form of variation. Let me erase my horrible drawings. All right, now we move on from prophase 1 to, guess what? Metaphase 1. Now, these chromosomes are going to be facing each other. So previously, before, remember they were lined up in one straight line? In metaphase 1, they don't line up in one straight line. They're on top of one another in one straight line. Okay, so we call that a tetrad. What does tetra mean? Do you guys know? It means what? It means 4. So there's 4 chromatids. See, 1, 2, 3, 4. So there's 4 of them in one section. So that's a tetrad. Oops. There we go. Something interesting happens here and in anaphase 1. So this chromosome right here could have easily got attached on top, and this one could have easily got attached on bottom. So just because you're looking at this picture right here doesn't mean that this has to be down here. So the chromosomes almost kind of randomly choose which direction they want to go. It's completely random. that random assortment of chromosomes has a name, and that also increases variation. So crossing over, where the chromosomes cross over and take pieces of each other randomly, that's increasing variation, but also that random assortment of chromosomes also increases variation. Okay, that's called independent assortment. Write that down. Independent assortment, right here. So independent assortment means these chromosomes randomly choose which direction to go in. So this one has chosen to go in this direction, and this one has chosen to go in this direction. But it's totally random. This chromosome could have chosen to go here, and this chromosome could have chosen to go there. It's completely random, and we call that independent assortment. We'll talk more about that when we get to genetics. Okay, next stage is anaphase. Okay, and there's your picture. So just like anaphase in mitosis, the chromosomes are pulled apart. And then the final stage is telophase 1. So now, at this point, we've gone from 92 chromatids to 46. Are you raising your hand, sweetie? Oh, what's up? Oh, restroom? Yes. So just like regular telophase and mitosis, you start to see the nuclear membrane come back, all that good jazz. It kind of looks like you're getting that cleavage furrow there. Okay, so I'm going to skip that because that's not that important. Okay, go to the next page. I'm going to get back to this. I'm going to get back to that. I want to go over crossing over real quick. So go ahead. If you've got two different colors, that'd be great. So here's my mother chromosome here, right? And it's already gone through interphase, so it's double stranded, so sister chromatids. Okay, this chromatid is the exact replica of that chromatid. And then here's dad. Thanks, dad. again, the chromosome I got from my dad has also replicated. So that is my homologous chromosome. During prophase 1, they cross over each other, just like that. This part right here is called the chiasma. So the point where they actually cross over is called the chiasma. This is sometimes also called synapses. Synapses. Crossing over, synapses is the same thing. Yes, you draw this. Please draw this. Crossing over and synapses is the same thing. And the point where they cross over is called the chiasma. It's right there. And so there's your under where it says crossing over? Yeah, is that not part of your notes? Yeah, I thought it was, right? Okay. And so this is your end result. This is totally random. I mean, we could have had this part remain blue, that part remain red, but this part have the red, and this part have, like, it's completely random which genes are going to get shuffled and switched. But here's what we know. This pair of homologous chromosomes looks different from this pair. Are we good? Does that make sense? It is no longer the same. And they are very different from each other now. This process happens differently in every one of your egg cells, ladies. This process happens differently in every one of your sperm cells, guys. So that means that every egg cell you have, ladies, has a different variation of your genes. And gentlemen, every sperm cell you have has a different variation of your genes. Like I said, if your first child comes out ugly, you have a second chance, a third chance, a fourth chance. I would suggest that after the fourth chance, you should just deal with the ugly children. I mean, because 4 children is a lot. But your children will all look different. Any question about that? Now, independent assortment says this chromosome right here could go to that cell, and this one could go to that cell, or check it, this one could go to that cell, and this one could go to the other cell. We don't know which direction each of these chromosomes is going to go in when we get to anaphase. Who knows? It's completely random. That's independent assortment. So that is another level of variation. So crossing over, independent assortment. These two are the ways that you get varied or different variations of genes in your egg and sperm cells. Okay, now I'm going to go back. Yeah, Jemana, the way that you get children that have no variation is you have identical twins. Hey, can somebody open that door for her? But identical twins happen when an egg and sperm cell fuse, and then the zygote splits. So they have the exact same DNA. That's why they look exactly alike. Write this down. Okay, now we're going to go on to meiosis 2. Okay, so now I'm going to go backwards because I skipped meiosis 2. So going down to meiosis 2. Skipped all that. All right, so you can skip interkinesis and go straight to prophase 2. In prophase 2, your spindle forms, nuclear membrane breaks down, nucleolus goes away, all that stuff happens. No crossing over. No crossing over here. We've already crossed over in prophase 1, so no crossing over. No, we're not doing that. Not again. If you can see that. No crossing over. No crossing over occurs. But for sure, the chromosomes in this cell and the chromosomes in that cell have a different variation of genes. So they're not the same. Not like mitosis where they're identical. These are not the same. So no crossing over, regular prophase 2. Metaphase 2, now we're in a straight line, like regular metaphase and mitosis. So no tetrads. So now we're just in a straight line. Spindle fibers are attached. God bless you. And then anaphase 2, sister chromatids start to separate, just like we normally do. Make sure you're writing down the events too. Don't just draw the pictures. Now you can start to see 4 cells being formed. You guys kind of see that? You can start to see 4 cells being formed. And then telophase 2. The chromatids arrive at their destination. You can start to see the nuclear membrane come back. And now you've got 4 cells. 4 sperm cells or 4 egg cells. Now, if you have egg cells, you're a girl, all your egg cells are available at the time of birth. So you have all the egg cells you're ever going to have attached to your ovaries at the time of birth. Okay? If you're a boy, sperm cells are constantly being produced. I think that's as much as it'll go. Hold on. Let me do it again. There we go. So your sperm cells are constantly being produced. Your prostate is what produces your sperm cells, and then your testicles, your gonads, are what hold the sperm cells in place until it's time for that magical moment that we're not going to talk about. Okay. So at this point, you went from 46 chromosomes now to 23 and 4 haploid cells. Make sure you write their 4 haploid cells. All right, so where it says meiosis overview, this is the meiosis overview you're drawing. So I'm starting here. I would say that this is, how many chromosomes do I have here? Can you guys tell me? 4. So I'm going to call this 4N. Does that make sense? Okay. They're double because you can see this one's a homolog of this one. They're the same color. Okay, so this is a diploid cell, and it is 4N. After interphase, after I go through S phase, they double up. After meiosis 1, they do that whole crossover thing during prophase 1, and they do that whole independent assortment thing during metaphase 1 and anaphase 1. So we don't know which chromosome is going to go into which of these cells. We have no idea. It's totally random. And then when we're all done, we have 4 haploid cells. They're either egg cells or little spermy cells. So let's say these are 4 new egg cells. Of course, I've got a little sperm cell that has to come along and fertilize it. The sperm cell could fertilize this one, or this one, or this one, or this one. That is your third level of variation. Who knows which sperm cell is going to fertilize which egg cell? So 3 ways you get variation in your children: crossing over, independent assortment, and random fertilization. It is totally random. You have no idea which one's going to fertilize which one. All right, next class, bringing in some gummy worms, and we're gonna play with gummy worms. It's gonna be great, and you can eat some gummy worms. If you bring gummy worms, I will give you extra credit. I will appreciate if you bring a baggie of gummy worms. So you'll get 10 points in your lab. On the back of your notes, you drew an egg and sperm cell, right? Please put random fertilization. It says fertilization on top. Put random in front of the word fertilization. So random fertilization. The bottom, we'll do that beginning of class next time. We'll also talk about what happens when chromosomes don't get separated correctly. We'll talk about that as well. So don't forget that your Jamboard is what is for homework. Yes? Only the Jamboard, yes. The Jamboard is what's for homework. If you have not completed other homework, you should do that. Next class, lab. If you bring your own gummy worms, Ms. Johnson will give you extra credit. Those of you at home, you will do something separate. Sorry, no gummy worms for you. Unless you want to do the lab and then you can go get some gummy worms and do it at home. It's up to you. All right, I am so done, and so are you, right? Which homework?
I think it was the DNA and replication homework.
You said you emailed it to me?
Yes, I've been emailed it to you. And it says I'm still missing the rotation. But I think
Which rotation?
I think you're doing with the I think it's the they're still on with the
Hold on.
the passive transport and active transport.
Hold on one sec. Hey, y'all. I'm going to end class now. Elijah, Jordan, Jumana, have a nice day. Jumana, I hope you feel better. Okay, bye-bye.